Alright, so I'm going to talk a little bit about endogenous and exogenous pathways of antigen presentation. And basically, antigen processing is just a immunological process that prepares these antigens when they come in encounter with special cells of the immune system called T lymphocytes. So it prepares them for this presentation to these cells. The process involves basically two different pathways for processing these antigens, and what they do is they process and can tell your self proteins, which are the organism's proteins, from the viruses, bacteria, any other pathogens that come encounter. So it has to tell the difference between those. The presentation of the antigens will be class 1 or class 2 of the major histocompatibility complex, or MHC. It's dependent on which pathway is used, depending on if it's endogenous or exogenous. Both of them require the antigen to bind to the antigen before they are expressed on the cell surface of the cell. So the MHC1 typically involves the exogenous or the endogenous pathway of antigen processing, and the MHC2 involves the exogenous pathway. Cross presentation does play a part in this, but I think it's normally towards the end of the endogenous pathway, in the latter portion of that pathway. So when I'm going to talk a little bit about each of them. A little bit about the exogenous and the endogenous. So the majority of the antigens that play a role in this are the exogenous antigens. These come from the outside of the body, and I'll have a picture in a minute to show the difference between the exo and endo. But certain, like, come through different infectious agents, like bacteria, fungus, protozoa, anything like that. Also from other environmental substances as well, like dust mites. Also, like what is another one? Pollen's a good example. Through 3 different forms of, you can get it through different ways: inhalation, ingestion, and injection. So those are ways that you can get those inside your body. They are taken up by the APCs, or antigen presenting cells, in the exogenous, such as phagocytic cells and dendritic cells, and macrophages are the phagocytic cells. Also the B cells, the B lymphocytes, which produce the antibodies. So the APCs, the way this works, they engulf the antigen by endocytosis. And I have a picture, which I'm going to show you in a minute, the difference between the two. It's just a little picture. So we see that in the exocytosis, it's being engulfed right in there. So you can kind of tell. It's engulfed by endocytosis. Endosomes fume with a lysosome. After that, the antigen is degraded into fragments, and this makes short peptides, and these peptides are displayed on the cell surface. Within the class 2 histocompatibility molecule, and they are recognized there by CD4 plus T cells there. So that's kind of how that works. And as far as the endogenous antigens go, they are generated within the body. Within the cells, through viral infection and intracellular bacteria. How this works is they are degraded into fragments, like I said, peptides in the cell, same as exogenous. They are then displayed on the cell surface within the class 1 histocompatibility molecule. Here, they are recognized by a CD8 T cell, and these are the cytotoxic T cells, and they can destroy the cells that are infected. Here, I have a picture here. So basically, we have here. This is the difference between the exogenous and endogenous. As we can see, the exogenous pathogens are here, coming from the outside. The antibodies and phagocyte activation by T helper cells is occurring here. That works by exogenous processing. The endogenous, right in here, pathogens, they are eliminated by the killing of the infected cells by CTL that use antigens generated by the endogenous processing. So that's a little picture right there. Shows you how those are inside, those are coming from the outside, and a little representation. I also have another picture. I don't think I can get it pulled up. Just shows that. But basically, like I said, the exogenous antigens are that's called endocytosed and presented in class 2, and the endogenous are presented in class 1. One's recognized by CD4, one's recognized by CD8 T cells. So that's a good little difference right there. Also, there's different stages of the processing: uptake, degradation, MHC complex formation, and antigen presentation are the different steps and different processes of that. And that's basically the gist of it.